Alignment-based methods. Alignment-based methods for motif discovery first construct a multiple sequence alignment of the set of sequences, where each sequence of amino acids is typically represented as a row within a matrix. Gaps are inserted between the amino acids so that identical or similar characters are aligned in successive columns. Once the multiple alignments are constructed, the patterns are extracted from the alignment by combining the substrings common to most of the sequences. One of the first automatic methods for the identification of conserved positions in a multiple alignment was the AMAs program, using a set-based description of amino acid properties. Since then, a large number of different methods have been proposed. For example, AL2CO calculates a conservation index at each position in a multiple sequence alignment using weighted amino acid frequencies at each position. The DeVos method is based on a statistical measure of the diversity at a given position. The diversity measures the proportion of the 20 possible amino acids that are observed. The advantage of the alignment-based approach is that no upper limit has to be imposed on the length of the motifs. Moreover, these algorithms usually do not need as input a maximum threshold value for the motif distance from the sequences. In general, this approach works well if the sequences are sufficiently similar and the patterns occur in the same order in all of the sequences. Unfortunately, this is not usually the case, and therefore most methods for motif discovery in protein sequences assume that the input sequences are unaligned. Alignment-free methods. The vast majority of motif discovery methods in bioinformatics are alignment-free approaches that do not rely on the initial construction of multiple sequence alignment. Instead, they generally search for patterns that are overrepresented in a given set of sequences. The simplest solution is to generate all possible motifs up to a maximum length L, and then to search separately for the approximate occurrences of each motif in the set of sequences. Once a list of candidate patterns is obtained, the ones with the highest significance scores are selected. This approach guarantees to find all motifs that satisfy the input constraints. Moreover, the sequences can be organized in suitable indexing structures, such as suffix trees, etc., so that the time needed by the algorithm to search for a single motif is linear in the overall length of the sequences. This simplistic approach has an evident disadvantage: the number of candidate motifs, and therefore the time required by the algorithm, grows exponentially with the length of the sequences. Computing a significance score for each motif further increases the time required by the algorithm. A number of more efficient tools have been developed to address these issues, and in the next lecture, we will discuss some of the more widely used ones.